Hi, Dr. Jansen. First of all, thank you so much for joining this interview. We have prepared a couple of questions and are looking very much forward to hearing your answers. Let's jump right into it. Could you please tell us a little bit about your background and your research focus?
Yeah, of course. First of all, thanks for having me. I am Mylene Jansen. I am a technical physician, which means that I combine medical knowledge with my expertise in more technology-related areas relevant to healthcare, such as medical imaging. So in my PhD, I used medical imaging to better understand processes that happen in the joint as a result of knee joint destruction treatment for patients with knee osteoarthritis. And now I am assistant professor at the UMC Utrecht, and my focus is imaging of joint degeneration and repair in musculoskeletal diseases.
Our focus on joint research is obviously overlapping, which leads me directly to our next question. Your team recently published a study using IBLab Koala, our knee osteoarthritis labeling assistant, to assess the association between radiographic changes and clinical improvements after knee joint destruction. Would you mind giving us more details about the study design and the conclusion?
So in that study, we evaluated 16 patients that were treated with knee joint destruction, and we compared radiographic outcomes that were assessed with IBLab Koala, patient-reported clinical outcomes, and changes in synovial fluid markers after joint destruction treatment. And we found improvements in pain and in Galvan-Lorentz grade and in joint space width, and those were all very nice to see, but not very unexpected because we'd already seen that before in previous studies. But this was the first time actually that we found a significant association between the change in joint space width as assessed with Koala and the changes in pain as experienced by patients, which usually we don't find this significant association. And we also found that the joint space width changes were associated with changes in synovial fluid markers, specifically MCP1 and TGF-beta. So our conclusion of that study was that automatic radiographic measurements with Koala showed improvement in joint structure after knee joint destruction treatment that actually for the first time showed a significant association with changes in pain and within synovial fluid markers. So that was very nice.
Yeah, those outcomes are exciting prospects. What impact do you think they could have in the future?
Several, I think. I think that these results could potentially help patient selection for other disease-modifying osteoarthritis treatments that, by definition, have to influence both structure and pain. They can also provide us with better clues to understand, hopefully, how exactly joint repair mechanisms work, because we know, for example, that knee joint destruction works, but we still don't really know how or why. So hopefully these results can help us point in the right direction for that. And lastly, I think that future trials could potentially also benefit from AI-based measurement methods to generate robust imaging analysis results that apparently have the ability to show associations with pain as experienced by patients.
Yeah, I totally agree with that. We also see AI as a tool for standardization and optimizing of resources in clinical trials without losing quality. ImageBiobsy Lab is currently gaining a foothold in the CRO business, and collaborations like the one with UMC Utrecht are a great way to, or example, to see how we can add value. What benefits were you able to draw from our collaboration?
Well, first of all, it was a very nice excuse for me to visit Vienna, to come visit you in Vienna. So that was very nice. But apart from that, it was a great chance to perform an objective evaluation of knee radiographs that we had in this study, because these specific radiographs were performed in regular care, which means that they were not performed according to our usual research protocol, which means that we couldn't analyze them ourselves with our semi-automatic method that we usually use. So without the evaluation by Koala, we could not have compared radiographic changes with synovial fluid marker changes. And these results specifically gave us additional insights into which markers and therefore perhaps with which pathways might be important to stimulate joint regeneration. So provided us with knowledge that we otherwise would not have had. And in general, it was a very nice and pleasant and smooth collaboration that in the end resulted in a nice publication in rheumatology. So that was also great.
Without spoiling too much, you and your team are currently preparing another study in which IBLab algorithms are applied and further developed. Would you mind giving us some brief insights on what we can expect?
Yeah, sure. We are preparing a study where we want to use IBLab's algorithm to see if we can use radiographs, knee radiographs, to, in a relatively early phase, predict development and progression of knee osteoarthritis. But as you said, we are still very much in the preparatory stages of this. So if you're interested, you're just going to have to follow our future updates on this.
Very excited about it. And yeah, we've come to an end. Thank you so much for taking the time.
Thank you.
I hope we will see you soon in Vienna again.
Definitely.